Welcome to today's USP PharmaTech webinar, mABs Biosimilars from Lab to Clinical. Thank you for taking time out of your busy schedule to join us. Each registration is unique and grants the right to the individual registrants only to view this webinar. Sharing this webinar with unregistered viewers, either during or after the performance, violates PharmaTech Associates' copyrights and is prohibited. Next. If you have not already done so, we ask that you take some time to read the PharmaTech Associates course disclaimer. You can download this presentation, including the disclaimer, from your USB education account. Please follow the instructions in your confirmation email to download the presentation. Next. We have two presenters today. Apurv Puri, Principal Engineer at PharmaTech Associates. John Akins, Biologics Practice Lead at PharmaTech Associates. Presenting today is Apurv. Welcome and thank you. Welcome and thank you to you both. I will now hand it over to Mr. Apurv.
So I'm going to be presenting sort of the lead, and then Apurv is going to work with me in sort of the color commentary of the presentation. So today we're going to talk a little bit about moving biosimilar monoclonal antibodies from the laboratory through to the clinic. Obviously, we have a great deal to go through. It's a complex process. We're going to talk quite a bit about how biosimilars are similars as opposed to identicals. And obviously the audience hopefully understands and appreciates that. The difference is actually coming into between the original reference monoclonal antibody product and those of a biosimilar are significantly different in terms of the way they develop from discovery and in through process development and then into the clinic. So we're going to touch on several topics here at the high level that are relevant to biosimilar production and development, which will include the obvious starting point in terms of cell line selection and culture process, the downstream purification, the analytical considerations, which actually turn out to be a significant effort when you're dealing with biosimilars, and then obviously some preclinical considerations and comparability. Just want to touch briefly on where we start in a laboratory from a discovery standpoint. There are really two general processes that have historically been used for producing monoclonal antibodies and, of course, biosimilars. The traditional fundamental and foundational approach is through hybridoma, whereby you're using animal sources generally as the source material for antigen injection and then antibody isolation and selection. More recently, since the advent of synthetic biology has emerged and evolved and become quite a bit more sophisticated, we've moved more and more into cell line production of monoclonal antibodies and obviously biosimilars. In this case, the process is well established in the laboratory, whereby we use synthetic biology to identify materials, clone them, screen them, and then insert them into production cell lines and hosts that we can then elaborate and then move forward into process development. Commentary that really starts the process here in terms of thinking about the differences between biosimilars and their reference drug products. If you think about industrial processing from the overall process, you can see when you go from a laboratory where you're focused on discovery and screening and identification, in the case of biosimilars, it's really a big effort to develop comparability between the reference drug and your monoclonal antibody biosimilar. But when you think about things coming out of the laboratory, the folks in process development and quality and regulatory have a whole host of other considerations to worry about. And let's talk about just one aspect of this for the moment, which is when I do my cell line development, I now have to expand that, and I have to expand that into a production mode and obviously downstream purification. In the laboratory, you're really just trying to identify that initial species and then trying to characterize it. In process and production, you're worried about things like contamination. As you can see in this slide, every time you touch the material, you're moving it into a different piece of equipment, or you're scaling it up larger. it enters into the opportunity for potential contaminants that could be microbial or viral in nature. You could also have genetic drift of the target material that you're trying to express. And so these considerations for bioprocess are significantly different than they are in discovery. Also, when you're talking about biosimilars, you have an additional consideration that a huge focal point is trying to draw the element of comparability back to the reference material that you're trying to work with. And so that also flavors in to a large degree how you move your process forward, the considerations you have from guaranteeing that you have a quality product, that it actually compares favorably or almost as closely as possible to the original drug product. Report of commentary on process development here that I haven't touched on. No? Okay, we'll continue to move forward. Okay, after you've actually made your product, you now have to isolate that raw bulk material and move forward and purify it. And this is an effort to try to remove adventitious materials, whether that be contaminants from the original host cells, that includes other proteins, DNA, cell debris, culture media, co-culture media, and waste streams that have come through that production process. First and foremost, and one of the biggest concerns for regulatory agencies is actually viral and microbial contamination. And so as you purify, it's not just good enough to remove byproducts or other cellular components as you isolate your target material. It's also the removal of potential pathogen agents as well. And there are well-established processes that are recognized by regulatory agencies, as well as industry standard for viral inactivation, including filtration, heat, and detergent processing. Once you've removed and validated the potential for contamination or the lack thereof, you can then do a polishing step, which allows you to isolate and have a result in high purity of your product. And then obviously a final cleanup step, which would basically guarantee that you've removed as much potential pathogens as possible. You then go through a formulation process and then a packaging process for your final drug product.
So John, if we take a step back here, you've kind of shown, at least I'm more familiar with the industrial side of things when it comes to monoclonal antibodies and biologics production in general. But it seems like the candidate isolation and those steps are a little bit unfamiliar. So as we kind of step through this presentation, are you able to expand upon some of the early phase kind of cell line considerations and selection things that both are required by guidance as well as kind of some considerations as you're picking the cell line, as you're picking the system to be able to produce the monoclonal antibody?
Yeah, so we're going to touch on that, actually start in the next slide. So if we think about going from the laboratory into the clinic here with biosimilars, one of the things that you have to consider significantly at the start of all this is actually the cell line development itself. And what you really need to focus here on is the design and the construction of your vector that carries the gene that represents your monoclonal antibody. Being able to screen through that through a variety of different techniques and synthetic biology manipulations allows you to identify a vector that has a robust nature in terms of the ability to produce under specific conditions your target material. In addition to that, what you really need to do is look at the transfection method to be able to insert the genes into your host cell line, and then be able to make sure that you can control those expression scenarios so that you can produce a high quality product under a robust set of conditions. In addition to that, once you've identified sort of the gene and vector and transcription or the transfection methodology, it's really then expanding and moving into the cell host itself. Very commonly now, CHO cells are a majority of the host systems that are used. Primary reasons for that is that they're extremely well defined. They're relatively easy to manipulate to get the types of production, post-translational modification, and purification. There's also a significant knowledge about its genetic lineage, and the ability to put it into a downstream process system to allow you to produce under robust conditions and manageable conditions. So once you've identified that cell line, then of course you have to move into, moving the culture media forward and really the cultivation conditions to be able to focus on titer and yield, as well as the biomass accumulation, and managing that fermentation process accordingly. The other considerations on this slide that we'll touch on very briefly really is, the idea of not only the bioprocess, but what ends up happening to the system once you get into, the preclinical trials and the characterization. One of the key issues that you have to realize with biosimilars, and it's important to recognize that it's really about comparability, and that being able to go back to a reference and say, hey, look, our molecule of choice is as close to, if not identical to, the original monoclonal antibody that's been forwarded as a drug product. And under those circumstances, this actually puts the onus on the biosimilar process development and discovery group to identify where that comparability is. And that goes through all aspects, from the structure to its function, its elimination, its safety, its immunogenicity, and so on and so forth. So let's talk a little bit about cell line selection. We'll start that at the front. So the considerations you have to think about here that go forward are really moving into cell line, the history, and we talked about CHO cells as an example, and we'll use them as the stepping stone here. Really understanding where they came from, their genetic lineage, how they've been handled, how they've been banked, understanding how their growth characteristics and their genetic proclivities are moved forward. Also looking at how they've been managed from the past to the forward, and how they would be used going forward into bioprocess. The other issues that you think about here is really production culture and how you test it. Really, we're worried always from a cost and benefit analysis of worried about yield and titer of the antibody, how many passages the overall bioprocess has, and that affects not only the quality of the product you make, but the stability of that product, as well as potential contamination elements. The other thing to think about as you think about bioprocess is really managing your CMC effort and getting lot-to-lot characterization to make sure that you get as low a variation as you can. Remember, these are biological systems, and so you can expect to see small variations. The regulatory agencies recognize that as well. Part of the reason biosimilars are termed as such is that it's a complex biological system that is almost impossible to get identity from lot to lot, and even against a reference standard. One of the key issues that we do want to think about too is also quality control, right? So how is my bioprocess set up? How is my cell line and my culture conditions set and documented so that we can guarantee that not only is the process robust and has good fidelity, but also we have a way to anticipate where there may be potential contamination elements, and also how those potential contaminating events are managed, and well, again, how are they documented, and how is that validated? Finally, when we start from developing cell lines, it's really how we store them. Keep in mind that the original progenitor cell that you've inserted your monoclonal antibody gene into really has elements of stability and history. And remember that the cell lines that you're developing are going to produce the products you want, but you also have to understand that they represent the history of your process, which is to say that you need to be able to compare back or have a reference back, not only to the original antibody that you're targeting as a drug, but also the antibody and the cell lines that you had historically in your bioprocess itself. So being able to store things with high fidelity, make sure that they're valid and robust storage systems. Being able to draw back to those storage systems is a critical element. Comments there?
And then, John, I guess the question for me on the quality control side, when kind of controlling sources of potential adventitious agents, what are some critical kind of considerations for your master cell bank, your working cell bank, and then your end of production line?
Right. So if we go to the next slide here, we talk about qualification of cell lines, and really for all intents and purposes, one of the biggest concerns that you really have to worry about is sources of contamination or opportunities for foreign matter to get into your process. And what's interesting about this, you start from the master cell bank, right? Which is to say that is essentially the source of truth for your process. And storing that under very rigorously controlled conditions is key. Now, obviously, when you want to run a process, you don't necessarily want to touch back into your master cell bank. You want to be able to have a secondary cell bank, which would be considered the working cell bank. That working cell bank is where you actually are moving from the laboratory or initial experiments and expanding that cell system into early production. That working cell bank is your reference point that you can draw from. And when you think about contamination from that point of view, obviously the highest threshold for making sure that you have a clean system from a contamination standpoint is the master cell bank, right? Because that's the source of everything you draw from. Interestingly enough, the number of passages that you think about from the master cell bank to your working cell bank determines to some degree how much rigor and how much testing you need to do to make sure that potential contaminants aren't carrying through in your process. So for instance, in sterility, that continues no matter what you're doing. Every time you touch something, every machine you put something in, every process you run has the opportunity for creating an unsterile environment. And so testing there runs the whole gamut from your master cell bank all the way to the end of your production. Other species like mycoplasma are similar. These are pathogens that can get in very easily in most locations, anytime you touch it, anytime you move it. Virus is similar, but really for all intents and purposes, the master cell bank, if it's clean, the working cell bank isn't necessarily required to test because it's one or two passages directly from your master cell bank. So the opportunity for contamination there is quite low. That's true for most of your viral contamination. And obviously, the last question to your point, Apurv, is cell line authenticity. Being able to track and identify and document the lineage of that cell line is critical, and that test should be done as well throughout the process that you run. We talked a little bit earlier about viral contamination. We've actually mentioned quite a bit of it, but how do you remove it once it gets in? There's always the opportunity that it does arrive, and being able to considering your bioprocess for removal is a key component of any bioprocess, right? So what we really want to think about is how does one test for this? We really want to make sure that a couple points are addressed. Number one, that it's quantitative. We need to have a very sensitive assay for viral presence, and more importantly, we want to be able to quantify just how much of it is there. We want to be able to understand how which viruses are there, so the identity, but we also want to be able to be very precise about how we design our experiments to test, so that we're identifying specific viruses that may be present along with other contaminants, and then not only how much is there, but how robust that method for testing is. Again, acceptance criteria for the test that you develop is critical, and being able to understand the statistical probability of contamination. So not only how much of it is there, but what is the chance and how significant of that risk is present at any given point. The other issue that you want to touch on here is really the variability between lots. We understand that every time you run a process, you're going to have slight variations, whether there was a conditional event that occurred, or whether there is just opportunistic changes throughout a process. Everyone should work very hard to make sure that those processes are consistent and reproduced to the level that they can, but variability does occur. What you want to do is make sure that you have assays that can identify that variability and quantify the results of that variation, and make sure that those tests and assays and the data that they generate is well understood, documented, and validated.
And then one additional question around viral clearance, John. Something I didn't see here are kind of, let's say I don't have kind of a baseline for certain adventitious agents in my process. Is the expectation that I am supposed to show that through some sort of spiking studies and show that I can demonstrate some sort of log clearance? And is that what we're talking about when we're talking about kind of this quantitation of viral reduction?
Right. So what the guidance suggests from regulatory agencies is that, in fact, you do run a process in which you use identified viruses, a select subset of viruses, as proxies or models to show how robust your identification process and your viral clearance procedures actually are. And so what you will do is the recommendation is to spike with those knowns and to demonstrate how effectively your assays are able to identify those, and more importantly, how effective your assays are at quantifying the presence of those viruses, and then subsequent bioprocessing steps, the ability to remove those viruses that may be spiked in. Talk a little bit about the downstream processing side of things here. The downstream purification process we had alluded to earlier really takes into essentially 4 high-level issues, right? Which is, think about the planning of your process design. How is the system going to be put together? How are you going to scale things up? How are you going to operate? What conditions are you going to think about in terms of the overall unit operations and how those unit operations are going to work together? As we've talked quite a bit about now, is focusing on contamination, particularly in biosimilars. One has to show that the product that comes out is as close as possible to the original drug product that you're trying to create. And as we mentioned before, the viral inactivation processes and procedures you put in should be planned in advance and well characterized. Fortunately, there are a great deal of examples of bioprocesses available and best practices that one can leverage. Finally, it's adventitious agents, right? So how do those agents get removed? What is the process by which they're being removed, and how do you characterize and qualify those? And that runs into a close relationship between your bioprocess group and your quality control group as well. It's important also that throughout the process, you make sure that you're retaining samples and you're using those sample retains as basis for analytical evaluation of in-process work, and also relative to your starting materials and your final products. Keep in mind that purification is supposed to result in a high quality product, but you need to be able to compare that purification process as you go through your unit operations in such a way that you're able to demonstrate that the product that you end up in the end is actually pure relative to what you started from.
So John, we've talked a lot about kind of cell line things and viral considerations and downstream things. But I guess the elephant in the room is, we haven't talked about the monoclonal antibody itself at all. So
Right.
what are some kind of, I guess, characterizations per regulations and other things that we really should focus on during the preclinical stage so that we can demonstrate that the monoclonal antibody is what it is, it does what it should, and ultimately is as pure as it needs to be.
Right. And so what's interesting here, as you're producing biosimilars, you actually have a slightly different mission statement, so to speak, than someone that's developing an initial drug. And what you have to deal with is you're always shooting for a target of the known, right? So if it was a new antibody that you're developing, the focal point there is to generate, identity for sure, but it's really clinical functionality. It's the potency, it's its stability, it's its immunology, its immunogenicity, or hopefully their lack of. But when you get into a biosimilar, it's all about how do I compare? How do I compare to that original reference material? And so there are a number of ways that you think about that, right? There are specifically chemical and biochemical processes that you use to actually characterize the identity of the antibody that you're producing, right? And that can run the gambit. And these are very significant undertakings, right? In the sense that you think about the antibody structure, not only from its primary sequence at the amino acid level, but its post-translational modifications. So if you're attaching carbohydrates to it or other structural variations. The other element here is that not only does it have the right sequence, but does it correctly fold, in the sense that does it have that secondary and tertiary structure that's required for function? Does that antibody actually aggregate, or does it stay well behaved in solution? These are key elements to identifying not only how it behaves how your product behaves relative to the reference, but how your product behaves when you start to do things like produce it and formulate it. Being able to draw parallels between the reference standard and your product, or comparability, is a key aspect and a major effort for biosimilar development. Now, there are a number of instruments that you can use, obviously, and there isn't really specific outline about what instrumentation or what techniques you specifically have to use. However, following best practices that would include, structural integrity, migrational patterns on acrylamide gels, the molecular mass, its proteomic identification, assays in terms of specificity with respect to, does the antibody bind to its target antigen as effectively and identically, or at least as close as possible to the original reference compound. Obviously, when you start testing functionality, you really need to have both positive and negative controls. And it really should allow you to get a very good picture of not only chemical similarity, but functional similarity to the reference. One of the things you want to look at here also to evaluate the biosimilarity is really the immunological aspects of things. So is your product actually creating a different environment when you assay it against either other opportunity tissues, so that we're looking at off-target effects? Do those fingerprints look very, very close to, if not identical to, what the reference antibody has? And then again, finally, what we said before, we'll revisit this here in terms of the characterization of the antibody itself, which is to say, you really want to make these assays and measurements as quantitative as possible. You really want to understand throughout the entire process whether your assays are sensitive enough and descriptive enough to give you a full picture of the monoclonal antibody biosimilar that you're trying to develop. Now we'll consider, go ahead.
Sorry, I guess this
Nope.
leads well into the next slide, because I feel like antibody drug conjugates have become a little bit more prominent. There definitely seems to be industrial attention that's being paid to them. So I guess I would like to learn a little bit more around kind of product characterization considerations that are specific to ADCs.
Right. So ADCs are a rapidly emerging type of product that includes antibodies and their associated components. One thing you have to understand about any antibody drug conjugate is that you're really dealing with two qualification, quality and regulatory principles. You have the antibody itself, and then you also have what is conjugated to it. If it's a small molecule, you're signing up for a small molecule regimen, whereby you're running an entire process validation of that small molecule. You're doing the same thing for your linker as well as your antibody. And so those elements all have to be treated in parallel and then ultimately brought back to the final product. And so we talk about the special issues about these incredibly valuable therapeutic agents. They also come with more complexity as well. You're running multiple validation processes because you're dealing with multiple products that are all aggregated into a single final product. And so when you think about how you manage these types of materials specifically, you're looking not only at whether you're making a quality antibody or making a quality conjugate, you also have to think about how they're joined together, right? Which is to say, my potency is linked to not only how my antibody binds its target, but the level of conjugate I have on that antibody that I'm delivering to a target. So for instance, if you're looking at an oncologic agent, you want to make sure that not only is that antibody recognizing well its target, but it's also carrying its payload. And more importantly, that payload is loaded to a level that we can accurately and qualification-wise determine its presence and its stoichiometry. Again, these are variables that become important when you start talking about end product potency and end product performance. Let's carry the conversation a little further into the analytical considerations of that potency and stability. So when you think about the potency assays here, you have a great deal of opportunity here to evaluate different methods and processes for determining potency. And again, what regulatory guidance would suggest is that you just have to establish that your biosimilar behaves like the reference compound, but more importantly, that it actually has a characterization profile from its performance, and we talk about potency in this case, that is quantifiable and reliable, and it's consistent, right? And so these can be looking at specific assays that are high sensitivity, and you can see these on the slide here. You can look at in vivo activity that is important for correlating the similarity comparability between your biosimilar and a reference compound. We talk about the ability for the material to be stable. So it's got to, it's got to get into a formulation. It's got to survive its formulation. It's got to survive packaging. It's got to survive its pathway to the clinic. It also has to be able to show that it doesn't crash out of solution or that it doesn't create other instabilities that would obviously, compromise the product integrity. Now, importantly here with all of these, you have to have a control strategy that allows you to understand the critical quality attributes that are involved with this product, and that includes its potency and its activity and its stability as well. And finally, I think it goes without saying at this point in the conversation that comparability is the critical aspect for all of this, and the onus of the sponsor to produce a biosimilar that replicates as closely as possible the reference compound that you're going after.
And then I guess, John, in the same vein here, we're kind of talking about maybe potency assays in general for mAbs, but then are there considerations for ADCs, then immunoconjugates that go maybe a step farther?
They do indeed, right? And so for immunoconjugates, as we've a little bit alluded to previously, these are much more complex molecules. And so you have two levels of potency and stability concern, right? That of the antibody itself that's recognizing the antigen that's trying to obviously treat a condition or disease. And more importantly, then it's also the conjugate itself, right? Is the conjugate stable in the system? Does it survive its transit from its point of infusion to its target? And then again, does it release properly with the right kinetics so that it can actually do its job as well? And so for all intents and purposes, you really have to deal with two chapters whenever you deal with immunoconjugates, right? It's not only how does the antibody behave, but it's also its payload as well. And again, you have to establish and demonstrate this through a robust CMC and quality program that allows you to identify, document, and validate that what you're doing here in these assays is representative of what you will need to generate for any kind of BLA filing or what have you, right? So more importantly here, we've talked a little bit about sort of the delivery side, but we haven't necessarily talked about what happens when it's in the body, other than saying, hey, look, it's got to be active. We have to look at how it behaves once it's done its job, which is to say, when it goes through the system, it gets cleared, it obviously has to be degraded. What are the kinetics in which that degradation and clearance occur? What are the potential side products that may happen in the process of clearing and degradation as the drug product is basically sunsetting in its use, right? Identifying also and quantifying the level of conjugated product versus unconjugated product. Remember, during the process, you're linking the conjugate to the antibody. And so, as understanding any chemical principle would tell you that you're not going to get 100% conjugation, identifying that and correlating that to its potential function and its potency actually is a critical aspect to qualifying and quantifying immunoconjugate products. Moving forward, talk a little bit about process monitoring and determination of consistency of your product, right? And so we want to be able to establish that we have a robust process to manufacture our biosimilar. In this case, we want to make sure that we are following through and tracking a lot-to-lot purity, a lot-to-lot sterility, a lot-to-lot stability. And so to be able to do this, we have several in-process tests that we look forward. The question, of course, is do we have to do all of these tests all of the time? And again, much like cell line qualification, we start thinking about the product that we're manufacturing. Of course, sterility, because we're touching that product in not only a number of manipulations throughout the process, whether it's coming out of the reactor, or it's going through chromatography, or it's going through viral removal. There's always the potential for a sterility issue, right? And that's a microbial and viral infection at any given point. So that is testing that is required at basically every step along the way, from the material that comes out of your bioreactor all the way through your final packaged product. When it comes to which types of contaminants are there, obviously we want to start from the bulk. That's obviously the highest threshold. Viral infections generally can occur at any given point, but the likelihood of them coming through the purified product is quite low, mostly because you're actually working hard to remove those species specifically. And the final product, it really shouldn't be there anyway, simply because that's the process that you've undertaken to clean the system up. In terms of adventitious materials like proteins like endotoxins and nucleic materials, generally here you're obviously going to have those potentially present in the bulk material, but through the purification process they should have been removed. And so the purified product for nucleic acids obviously would want to be tested. The final product doesn't necessarily, if it passes through purified product, the final product should also carry a similar purity. So testing isn't necessarily required there. In the endotoxin process, the purified product and the handling aspects of it, that endotoxin may or may not be removed. And so testing that really is important at the final product, but not necessarily some of the upstream elements.
And John, I guess one question here, let's suppose I'm unable to complete certain types of testing on the bulk, either due to interference issues or otherwise. Are the agency's expectations then I identify that in some sort of a risk assessment and then prepare for some kind of testing upon the final product and set that as a release spec? Because I would imagine not everything works out as neatly laid out by the guidance. And so in those situations where certain things may not be completely in line with guidance, what would you say is kind of some salient advice to make sure, one, the risk is low, and then two, you're at least demonstrating to the regulatory bodies that you're doing what you can to keep the sterility assurance high and the risk of contamination low?
Yeah, I think the issue there, Purv, is how you set up your assay protocols, the robustness by which those protocols are evaluated, and then again, the documentation and the validation of that documentation are key elements to the narrative that you have to present in your package, right? And so essentially, A, using best practices, B, making sure that those best practices are documented, and then third, make sure that those documentation elements are robust and validated in your process. And so that represents an opportunity for you to the best that you can. And again, it's best practice, right? And so as always, things are continually evolving, processes are getting better. And again, it's really showing that that chain of custody is a key aspect of that element. I want to talk a little bit very briefly about drug product stability. We've talked quite a bit about this, but really here what we want to deal with is the guidance here is to make sure that you prove, and this is true for quality assurance and bioprocess documentation and testing, to make sure that your product is what you say it is, right? And so that goes through the chemical assays you look at, the biochemical assays you look at, and then also the qualification protocols that you've put into place. We want to talk a little bit about the preclinical considerations. Now, obviously, we're going lab to clinic, so we're getting close to the end here. And really, at this stage, we want to look at the comprehensive elements of the data package that you're putting together, right? And so not only do we want to characterize our product, but we want to look at its performance, right? And it goes through in vitro assays that determine reactivity. Is it hitting the target I want, or is it hitting a bunch of artifactual tissues that create off-target effects and potential hazards for us from either a reactivity standpoint or a toxicological standpoint? With here, this is where you start seeing not only in vitro work, but also animal toxicology studies. There's obviously some debate about the value of animal models at this level, but it does give you a sense and a reasonable proxy to determine what your dosing regimen is and the acute toxicity, clearance rates, and half-lives, particularly if the animal model is well set up. And again, we want to look at in vitro work in the laboratory to establish that the pharmacodynamics and the behavior of that molecule that you're putting into the system as a drug product represents the performance, potency, and whatever of the original reference compound. So pharmacokinetic studies, I think many of you in the audience are familiar with what goes on here. Again, it's all about comparability. That's the theme that we want to drive home here. We want to determine all of the functional characteristics and move things forward through the development process in such a way that this represents the reference standard as closely as possible. Assays should be similar to or identical to the original reference compound. Dosing and immunoconjugate, if you're using an immunoconjugate, for example, that the whole molecule behaves as it should. When we start thinking about clinical considerations here, right? So this is, right now the handoff into the clinic. Up until this point, we've really wanted to show comparability. But more importantly, we also want to show that performance and material that we're working with here is safe. It's formulated in a stable sense as well, right? And so this gets us set up for consideration of dosage and mechanism of action. We need to show that the mechanism of action and dosage is representative of the reference compound as well. And we have to establish that proof point is in fact true and valid. Comment a little bit about dose setting here. The in vivo dose has to be based on both in vitro and the antigen antibody affinity. We have to establish that through bioassays as well. One of the things to consider if you don't have good solid animal models available, we really want to introduce the dosage setting at the lowest possible level to determine at the tissue culture level where those potential challenges are and identify where potential toxicological elements or metabolic processes are going forward with that. Comment about assaying immunogenicity, right? And we talked a little bit about this before, but we'll revisit it for a moment. Number one, we want to make sure that our study comparisons are appropriate and valid. which is to say we always want to go back to our reference material and compare performance of that reference material against the biosimilar. We always want to have positive and negative controls. We want to identify epitope specificity to make sure that we're actually targeting the right part of our antigen that we want. And we want to make sure that we're mimicking standard curves of performance in terms of immunologic response and on target binding. Again, as always, it's really important to have assays set up that are precise and sensitive to make sure that the data that we generate is representative and can be drawn back to the original reference material.
And I guess, John, next question's for me around this. So now that you've kind of laid the ground for at least the preclinical and clinical considerations overall, if I were to compare, let's say, an innovator product to biosimilars, what am I looking at as far as kind of my burden for IND and kind of I guess this slide would be a good one where, you know, how do you demonstrate how the data requirements are different? And then I guess what are you've already answered some of the key questions around, I guess, comparability. Seems like potency is very important. Demonstrating immunogenicity and serum half-life seems to be pretty important. Kind of
Right.
what are some other considerations you would give for kind of demonstrating this comparability? And actually, ultimately, you know, what does the agency require for something like this?
So the agency let's start from that and go backwards, right? The agency requires that you have a comparable product, right? And that includes not only how it's characterized physically, you know, biochemically and performance-wise, but it also compares to how your process sets up, right? How your studies and your risk management plan are put together. Everything for a biosimilar is targeted to comparison, right? Whereas your original drug product, it obviously has its risk management plan and its clinical studies and its qualification and characterization. But from a standpoint, it's the icebreaker molecule, right? Which is to say it's first in its class. The biosimilar, on the other hand, has an additional burden to not only show its performance as a drug, but that its performance as a drug as closely as possible mimics the performance of the reference drug as well. And so many ways they're similar in terms of the absolute data that you're collecting. Obviously, the robustness and the quality performance have to be well documented and valid. But more importantly, on the biosimilar side, it's that extra burden of comparability that you have to deal with, right? And that comes through quality and process as well as performance and characterization. All right. So again, in terms of if we look at this slide here real quickly, you know, we talked about how do we show comparability, right? And so if we talk about characterization, and this is really a sort of summary slide, you know, it's really the laboratory analysis, the biochemical characterization. We talk about pharmacokinetics, otherwise, you know, performance. That's a combination of comparability between the in vitro and in vivo assays to show that your biosimilar is, in fact, as close as possible to what the original reference is. You know, when you get into early clinical studies, it's showing that, you know, the performance of the material is as close to possible, you know, in terms of its toxicity profile, its pharmacokinetics and dynamics, and also how it clears. From a safety standpoint and efficacy, these actually show up in the clinic. And again, safety itself is a primary element and a key aspect, not only all drug products, but biosimilar specifically, because the safety profiles, as well as everything else, needs to be as close to the original reference drug as possible. Finishing up here, we'll talk a little bit about what we've gone through here in the end, which is we've touched on cell lines, the analytical methods and stability, the preclinical, some of the clinical aspects we didn't get much into, and then the comparability and biosimilarity, all of which really is the key aspects to biosimilars is the comparability, right? It's how does your drug biosimilar compare to the original reference? And it's how it's physically similar, how it's produced, and what the production process gives you, how the quality system is set up, and how it's created a valid narrative for how your drug is not only created, produced, purified, and then ultimately packaged, but it's also how we've shown that that process is producing a similar to reference as possible from structure and function. Further conclusions, right? So when we start talking about moving the monoclonal antibody biosimilar from its lab to clinical, obviously these are complex products. We have to deal with a very involved process for analytical characterization. We really also want to make sure that we're communicating and planning early in the process to establish how the various unit operations, bioprocess, quality, regulatory, analytical, can work together and hand off their product from each work function so that you're informing the next work function, but also able to create a full and comprehensive and understandable package that you'll ultimately submit for approval. And again, not to mention this too many times here, but we have throughout this presentation, comparability is the key aspect to a biosimilar, in the sense that you need to show those elements, you need to prove those elements, and then you need to show that you have control over all of those elements as well. And with that, we'd like to end our presentation here and answer questions if we have time for them.
I guess, John, I can probably take the early phase considerations question if
Sure.
you're good with that. That's at
Mhm.
least in my wheelhouse.
Hmm.
So as you're at least going from kind of lab to your first clinical batches, especially as you're changing scale and systems, it's pretty important to understand kind of the underlying engineering principles for culture. So your lab process may very well be based on some sort of shake flask method, and it's, producing generally decent titers, but you may or may not be able to produce more than, a few doses or so with that. So as you kind of move up to larger systems, it's it needs to be pretty clear that your cell line is essentially experiencing the same conditions from, let's say, the shake flask to the final suspension culture. And sometimes that can be pretty difficult to demonstrate, but ultimately, if your passaging is stable, you're controlling at the right temperature set points, your cell line's not hypoxic, and there are no indications showing that, and then your antibody titers are at least the same, I think that's a pretty big first demonstration that you're seeing the same conditions, or at least your cell line is seeing the same conditions at those larger scales. And so it's pretty important in those first studies that you characterize whatever system you're going into. So, and by system, I mean whatever bioreactor you're going into. A lot of vendors these days will offer quite a bit of characterization data on their bioreactor systems. And then it's really important to understand kind of the shear forces that are generated at the impeller tip, kind of the overall considerations for, dissolving air into the culture to make sure the cells are getting what they need, they're as productive as possible, and you're getting to the optical densities that you need to be able to get the right antibody titer. And so I think those are just some of the considerations, but there's a whole basically kind of underlying theory for proper cell culture scale up that really should be followed. I think at this point it's basically been characterized and explored fairly well. And so for people kind of looking into that, it's very good to have an understanding both of the lab side of the methods and then having an engineering team that really understands how to specify the right user requirements and the right settings and the right engineering studies and the right level of analysis to be able to demonstrate that the conditions are the same.
Yeah, let me take a crack at the formulation one. How early should you consider formulation? I'll make a couple comments. One is, number one, you want to be able to manage your process overall, which is to say, from a techno-economic standpoint, you want to make sure that overall you're working as efficiently as possible. And so that includes multiple groups, your quality group, your bioprocess group, your analytical group, your, discovery, what we'll call discovery group, which is, the laboratory research side of things. I think once you consider formulation as early as you possibly can, which is to say, when you have a biosimilar that you feel comfortable with, that you can actually manufacture in a reasonable scale, so that you're starting to think about um, you've characterized the material, you're comfortable that the material you're producing is in fact a quality biosimilar that replicates, for all intents and purposes, its chemical and its physical and its biochemical processes. And I think at that stage, you could start thinking about the formulation elements. Until you have a material, a biosimilar material that is in a state that is chemically representative of the reference, that it has a stability, and that it is pure enough, you really shouldn't think too much about formulation, just because you're going to see variations and you don't want to have to go through formulation processing over and over again. So I think, you want to try to get it as early as possible, but it would be really, prudent to think about once you have that, have that agent to a level where it is essentially representative of the reference, then I think you should go ahead and start going with the formulation and dealing with the variations in process conditions that a formulation has.
And then I guess for this kind of, let's see, for the next question. So the regulatory expectation of similarity for the process and control strategy. I'm not aware of a specific guidance, but we can certainly look into that for you. As far as kind of the regulatory expectation of similarity with the process and control strategy, I mean, at the end of the day, your process and control strategy just needs to be able to produce the product that is going to be able to meet all your kind of final release specifications. And then ultimately the release specifications are in some ways related to the original innovator product. But in my mind, as long as your process control strategy produces what it's supposed to per your release specifications and in process specifications, I don't know if there's really any kind of expectation that those need to be exactly similar. And so if, let's say, within your process control strategy, when you scale up, your sparging rates might be different than what's in the filing for the original innovator product. I don't know if the FDA will necessarily see that as a difference in the way you control your process, let's say cell culture. So I think in that way, I don't know if there is any specific expectation or really any guidance that would say your process control strategy needs to look the same as the original innovator product.
Yeah, and I think the one thing you have to understand too, right, is if you think about the timeline of development, time is moving forward all the time. Technologies are improving. So for instance, many, many, many years ago, when some of these early drugs that are, coming off patent and are eligible for biosimilar treatment, automated processing equipment may or may not have been available. Some of those advanced technologies may not have been available or well adopted. They may not have been applied to the original drug agent. And so to your point, Apoorv, I agree with you. The control strategy and the process used to develop your product is obviously, it's important that you document it and that you demonstrate that CMC effort results in a product that can be characterized and qualified, and more importantly, that it is reproducible and consistent. From that standpoint, I think that's the bigger issue here, that the drug that results is something that is representative.
And I guess we're running out of time here, so I'll try to combine maybe some of the clinical questions together. I think for biosimilar clinical studies, is it acceptable to conduct phase 1, phase 3? And then for biosimilars, which clinical phase can be omitted? In my mind, this is not so clear. I mean, at the end of the day, the FDA is expecting an adequate clinical PK and PD comparison between the proposed biosimilar product and the reference product, and really a pretty detailed clinical immunogenicity assessment. And so, what I would expect for a biosimilar are essentially what I would just deem as like additional clinical studies that look detailed into clinical pharmacology. Phase 1, phase 2 are really going to be the safety piece, but these additional clinical studies, like they really can't be considered phase 3 or pivotal in my opinion. So those are necessary for the standalone development of an innovator product. But as far as biosimilars are concerned, your clinical just really needs to demonstrate the same level of safety, efficacy, and at the end of the day, really similar clinical benefits. So I don't know if it's as clean as we can just skip these kind of clinical steps. I mean, what's your
Right.
opinion there, John?
Yeah, I mean, I think that's one of those elements where, we haven't really said this, but I'll say early and often is important in terms of interacting with the regulatory body. Get a relationship with the regulatory agency, understand, present that, present your plan, present your intentions, particularly in the biosimilar space, and make sure it's probably much more important that you have a clear pathway to demonstrating comparability than anything else. And I think
Yep.
from a clinical trial standpoint, again, these are case by case cases because one modality or one therapeutic agent may have a very, very different profile, even from the innovator standpoint, and it may require closer scrutiny. And again, I think the agency can provide that, that insight as well. And so I think, to your point of Purv, just skipping over the expectation that you can short circuit some of these issues is probably something you should be very careful about.
Yeah.
As we're sort of out of time, I have one comment about because it's a pretty short answer, comparability and preferred number of batches. You really are looking at least 3. That's sort of the industry standard. That's true for small molecules. It's true for large molecules. You have to have a statistical, basis for comparability, and that is not only in terms of characterization, but also the process that you use to make it.
Yeah, and I would argue like sometimes n equals 3 may not be enough. At the end of the day, like John said, you really need to tell me what your statistical acceptance criteria are to be able to detect a difference between the innovator product and the biosimilar. And so if your statistical studies are not adequately powered enough, I think the agency will certainly push back on that. I think gone are the kind of easy expectations from maybe 30, 40 years ago, where you could demonstrate through a small number of batches. But I think sometimes being able to statistically detect a difference may require significantly more than 3 batches.